Hello. Today I'd like to cover Chapter 2, the chemistry of life and the origins of cells. All animal functions and behaviors have chemical bases. While chemistry is a separate field of science, its principles form the basis of biology. As you learned in the last video lecture, all living things demonstrate hierarchical organization. You cannot truly understand the higher levels of organizations, such as organisms or ecosystems, without understanding their component parts, like molecules and cells. However, because the topic of biochemistry is a deep subject in itself, this lecture will be limited only to the discussion of water and the major macromolecule, carbohydrates, lipids, proteins, and nucleic acids. This will, this will also allow us some time to talk a little about chemical and cell evolution. The items for discussion in this lecture will cover water among the inorganic substances in animals, followed by a discussion of the major macromolecules of the animal body, and then the rest of the discussion will be about chemical evolution and cell evolution. Elements are the most basic unit of matter. They can't be broken down in chemical reactions to any other constituent substance anymore. Of the 92 naturally occurring elements, animals need 25 elements in varying proportions. But only 4 elements make up 96% of living matter, namely oxygen, carbon, hydrogen, and nitrogen. Oxygen, as you will find in this table, takes up the highest percentage in the body. What do you think accounts for this high percentage of oxygen in the animal body? You can add this up to your assignment later. Water molecules contain 2 hydrogen atoms bonded to 1 oxygen atom. The large oxygen atom has a stronger attraction for electrons than the small hydrogen atoms. This unequal pull on electrons makes oxygen atom slightly negatively charged and the hydrogen atom slightly positively charged. This unequal sharing of electrons is called polar covalent bond and results to polarity of the water molecule, or dipole. This also causes the hydrogen atoms of one water molecule to be attracted to the oxygen atom of another water molecule and vice versa. Water can change from a liquid to solid and from solid to gas and back, but its molecular structure always remains the same. Water has some unusual properties due to its hydrogen bonds. Pure water is tasteless, odorless, colorless, and transparent. But due to the polarity of the water molecules, water is highly cohesive. This is the tendency of water molecules to stick together. Yeah, water also has a high surface tension, which is due to the cohesive forces of water at the surfaces. The water at the upper surface of a body of water usually is strongly attracted to the water molecules below it, forming a compact, relatively flat surface. And a lot of insects and animals take advantage of this compactness of the water surface, like water striders or spiders, and even the basilisk lizards that run over the water when they are running away from predators. Adhesion is also another unique property of water, which is due to water molecules sticking to another surface that is polar. This can result to capillary action, wherein the water molecules sort of creep up against the force of gravity in narrow spaces, like in a tiny tube. Small insects like sea roaches and thorny dragons take advantage of this capillarity when they draw food or liquid water on substrates. The water's boiling point is at 100 degrees Celsius, which makes water uh, unable to evaporate easily and leaves water available in many places on the planet Earth. This keeps our ocean and rivers full of water. They do not easily evaporate, unlike other kinds of substances like ammonia. Water also has a very high specific heat. That is, it takes a lot of energy to raise the temperature of 1 gram of water. And this specific heat and that of the boiling point of water enables many animals to regulate body temperature by regulating the distribution and evaporation of water from their bodies. When we perspire, for example, we remove a lot of heat from our bodies because of this specific heat, and so we release a lot of heat and therefore lower our body temperatures by perspiration. The melting point of water is 0 degrees Celsius. Below this temperature, water is solid ice. Unlike most chemical substances, water in a solid state has a lower density than water in liquid state. This is because water expands when it freezes. As a result, water molecules are spaced further apart in ice, giving ice a lower density than liquid water. A substance with lower density floats on a substance with higher density. This explains why ice floats on liquid water, whereas other substances or solids sink to the bottom of liquid water. In a large body of water, such as a lake or the ocean, the water with the greatest density always sinks to the bottom. Water is most dense at about 4 degrees Celsius. As a result, the water at the bottom of a lake or the ocean usually has temperatures about 4 degrees Celsius. In climates with cold winters, this layer of water, 4 degrees Celsius water, insulates the bottom of a lake from freezing temperatures. Lake organisms such as fish can survive the winter by staying in this cold but unfrozen water at the bottom of the lake. Organic molecules that make up the bulk of the animal body consist of long chains of carbon and hydrogen called hydrocarbon. The carbon atoms form covalent bonds with each other to form the backbone of the molecule. Although carbon atoms make up the backbone of organic molecules, other elements such as hydrogen, oxygen, nitrogen, sulfur, and phosphorus are also attached in small groups to the carbon atoms, and these are called functional groups. Here you can see in this structural configuration of lauric acid, you have the long hydrocarbon chain, and then you have the small functional group, in this case a carboxyl. Functional groups of macromolecules are specific groupings of atoms within molecules. They possess their own characteristic properties that determine the properties of the entire organic macromolecule. This includes hydroxyl, methyl, carbonyl, carboxyl, amino groups, phosphate groups, and sulfhydryl groups. The functional groups determine the shapes of macromolecules, and this in turn determines their functions. Functional groups can affect the bonds that hold the macromolecule together. Functional groups can also affect the polarity of the bonds. And finally, the functional groups add, uh, can contribute to the interactions of the macromolecules. In this slide, I just want to drive home the importance of functional groups, showing the two sex hormones of animals, testosterone and estradiol. Testosterone is different from estradiol mainly by a methane group that is shown in the red arrow. The difference between these two hormones by a methane holds the very big difference between a male and a female. Given the diversity and complexity of life on Earth, it's surprising that the most important macromolecules found in animals can be grouped only into four classes. These are the carbohydrates, lipids, proteins, and nucleic acids. The structural chemistry of the macromolecules play an essential role in its functions. Like water, they exhibit unique emergent properties due to their atomic constituents, as was mentioned earlier. Let's start with carbohydrates. Carbohydrates are polymers of simple sugars or monosaccharides. They are compounds made up of atoms of carbon, hydrogen, and oxygen in a ratio of 1:2:1. You have a glucose unit here that's shown, which is made up of such ratio: 6 carbon atoms to 12 hydrogen atoms and 6 oxygen atoms. C6H12O6 as the formula denotes. The carbohydrates are classified as either monosaccharides, which are mainly made up of simple sugars, and the disaccharides made up of 2 units of simple sugars, and then the polysaccharides made up of more than 2 units of simple sugars. Monosaccharides includes glucose, fructose, galactose, ribose, deoxyribose, and dihydroxyacetone, whereas disaccharides forming sucrose, lactose, and maltose. Sucrose is made up of units of glucose and fructose, whereas lactose is made up of glucose and galactose, whereas maltose is made up of 2 units of glucose. The polysaccharides, starch, glycogen, and cellulose, are made up of several units of glucose molecules. Starch is the storage polysaccharide in plants. They are made up of 2 kinds, amylopectin and amylose. The amylopectin are the branching form of starch, and amylose is unbranched. Glycogen is the storage polysaccharide in animal cells. They are stored largely in muscle cells and liver cells. Glycogen, however, does not last long compared to starch because they're not stored in very large quantities, so they have to be replenished almost daily. Cellulose is the polysaccharide that makes up the walls of cell walls in plants. Lipids are compounds that are mainly made up of long chains of carbon and hydrogen atoms linked by nonpolar covalent bonds. Because of these nonpolar covalent bonds, the lipids are characteristically hydrophobic in varying degrees. Some lipids are simple lipids, like these chains of hydrocarbons, the fatty acids. In this example here, you have a saturated fatty acid, which are long chains of hydrocarbons with all their carbon atoms fully bound to hydrogen. So they form a straight hydrocarbon chain. And here you have a fatty acid that has a couple of its carbon atoms that are not bound to the full number of hydrogen atoms. So they form double bonds between each other. And because of this, there's a space in this point wherein the molecule can bend, forming a kink. This is called an unsaturated fatty acid, and the kink or the bend is characteristic of unsaturated fatty acids. Because of these kinks, the fatty acids can occupy more space when they are held together, and because of this greater space, it makes the collection of lipids more liquid. A good example of this is olive oil, made up of unsaturated fatty acids, which is liquid at room temperature. Among the saturated fatty acids, a good example would be butter, which is compactly made up of fatty acids that are compactly arranged, and so forms solid lipid or solid butter at room temperature. You also have glycerides, as shown here as another classification of lipids. The glyceride that is shown here is a triglyceride, or fat that is stored in fat cells. It's made up of glycerol that is bound to 3 fatty acid tails. 2 of the fatty acids are saturated while the third is unsaturated, and so forms a kink that makes the whole triglyceride liquid even when stored compactly in fat cells. Then you have a complex lipid here shown. Complex lipids are formed by special kinds of polar molecules that are bound to fatty acids, hydrophobic fatty acids. As a result of this, in this example, it's a phospholipid. The phospholipid part makes a hydrophilic head, which is attracted to water molecules, and then the fatty acid tails is hydrophobic, and so forms a hydrophobic part of the entire molecule. The phospholipid therefore exhibits 2 properties, a hydrophilic head and a hydrophobic tail. This is very characteristic of phospholipids, and when they are immersed in water, such as in the water environments of the cell, they form 2 layers, wherein the phospholipid hydrophilic heads are facing the watery environments, either interior or exterior surface of the plasma membrane, and the hydrophobic tails insert themselves into the inner layer of the membrane, which is devoid of water, and so they form a double layer phospholipids that make up the plasma membrane of cells. The third classification here is steroid, made up of 4 fused rings of carbon atoms. Good example of steroids are the sex hormones that I showed earlier in a slide, testosterone and estradiol. And then cholesterol as well is formed by steroids. Cholesterol is also a very important component of the plasma membrane that makes the plasma membrane fluid as well. Major functions of lipids includes energy storage, like that served by triglycerides in fat cells, chemical messengers such as that served by steroids forming the hormones. And then as structural lipids, you have the phospholipids that make up the bulk of plasma membrane in cells. And then some lipids are also used as natural coatings, like waxes and oils on the surfaces of animal body and parts. The proteins are polymers of amino acids composed of central carbon atom bound to an amino group and to our carboxyl group and to a hydrogen atom, and finally to the radical group or the R group. This is the distinguishing group of atoms of each different type of amino acid, the radical group or R group. So every different amino acid have different R groups. Most proteins are the most abundant and diverse macromolecules in the animal. Proteins are made up of just 20 kinds of amino acids, but the possibility of arranging these different kinds of amino acid can be very diverse. Amino acids are linked together in covalent bonds we call peptide bonds by dehydration synthesis. And so here when you bind two amino acids together by dehydration synthesis, you form a dipeptide. In this chemical reaction of synthesizing a dipeptide, you lose a water molecule from one of the amino acids. And so we call it dehydration synthesis. The amino acids, because of their variable R groups, exhibit different properties. Some amino acids are hydrophobic, some are hydrophilic, some have charged R groups, and so they can be hydrophilic, or they can attract other charged molecules. Some are neutral. Now, because of the diversity of proteins, they can serve several different functions. Many proteins can serve as enzymes. Some are structural proteins like keratin. Some proteins serve to transport other commodities like hemoglobin, for example, that binds and transports oxygen and carbon dioxide. You have storage proteins like myoglobin that stores oxygen in the muscle cells before they are used for energy synthesis. And you have contractile proteins in the muscle cells, myosin and actin. Then you have defense proteins such as antibodies, melanin for pigments, and even the toxins or venom of snakes. These are composed of defense proteins. And then finally, some proteins serve as signal proteins, like many of the hormones that we have in the animal body. Proteins have very complex three-dimensional structures. To make analysis of the protein easier, we often take a look at them at 4 levels of structural dimensions: the primary, secondary, tertiary, and quaternary structures. The primary structure refers to the sequences of amino acids in its polypeptide chain. The secondary structure is based on the bending or coiling of the polypeptide chain due to hydrogen bonds between nearby amino acids. This results to either an alpha helical structure of the polypeptide or a pleated beta sheet. The tertiary structure is the overall three-dimensional configuration of the polypeptide, which is due to the interactions of the R groups between distant amino acids. These interactions among distant amino acids may include hydrogen bonding, ionic bonding, dipole-to-dipole interactions, London dispersion forces, basically the whole gamut of non-covalent bonds. Finally, the quaternary structure refers to the combination of polypeptides exhibiting the above-mentioned levels of structures. So proteins may be combined together to form quaternary structures. Many of the important proteins that we know of, like hemoglobin, are actually made up of different polypeptides that are at tertiary structures, and so they form quaternary structure. Proteins may be very complex and often stable, but when they are exposed to change of pH or heat, they can lose their high order structure and become denatured. Do you remember this molecule? I showed this in the first chapter lecture video. Um, with this change in just one amino acid, serine, the molecule luciferase changes its light emission into red. What do you think would happen to the firefly with regards to its reproductive success if it cannot emit the right kind of light? Nucleic acids are polynucleotides. That is, they are long chains of nucleotides that serve as building blocks. A nucleotide is composed of a nitrogenous base, a pentose sugar, and a phosphate group. In nucleic acids, the pentose sugar of one nucleotide is bound to the phosphate group of another nucleotide, forming the sugar phosphate backbone. And the nitrogenous base projects perpendicular to the sugar phosphate backbone. The nitrogenous bases come in two forms, pyrimidines and purines. The pyrimidines includes cytosine, thymine, and uracil, whereas the purines includes guanine and adenine. In the DNA, the nucleotide bases includes guanine, cytosine, thymine, and adenine. In RNA, it includes guanine, cytosine, adenine, and uracil. Uracil replaces thymine in the RNA molecule. There are two major forms of nucleic acids in the animal cells. This includes DNA, which is shown here in this figure, composed of two long chains of nucleic acids that are intertwined around each other with their nitrogenous bases held in hydrogen bonds, and their sugar phosphate backbone form the rungs of what looks like a ladder, a helical ladder. The RNA is composed mainly of a single chain of nucleic acid, but this single chain of nucleic acids can form hydrogen bonds of their nitrogenous bases with non-adjacent nucleotides, and so they can form loops. They can form shapes like a clover leaf in the cell's natural setting. DNA is replicated before cell division. Prior to the DNA chromosomes is doubled up in preparation for their segregation in daughter cells when the parent cell splits into two. Each of the two strands is used as a template for synthesizing new DNA, so that at the end of DNA replication, each resulting DNA will have both old and new copies of the DNA strand, a semi-conservative state of replication. Of course, this happens with the help of enzymes that bind to the DNA. This will be discussed in further detail in the latter chapters. The story about chemical evolution, that is how complex molecules of cells evolve, is a complicated story to tell because the reports or evidences are fragmentary and are not synthesized into one big story. But definitely the process of chemical evolution, that is the formation of complex molecules that led to the formation of living cells, are believed to have happened in three stages. The first, the formation of simple molecules in the primitive Earth's atmosphere, like ammonia, water, and methane, because of the combination of elements or atoms by the um, energy-rich atmosphere, because of a lot of atmospheric events such as lightning, UV radiation, and heat, and then followed by the synthesis of simple organic compounds. This was given evidence by Stanley Miller's classic experiment in 1953, when he subjected simple molecules like ammonia and water and methane to simulated bolts of lightning by generating electrical sparks in a vacuum containing those simple molecules, which resulted to the synthesis of complex molecules like amino acids and fatty acids. And then you have the formation of self-assembling proteinoid microspheres called protocells. That's the third event. This third event probably led to the development or evolution itself of the prokaryotic cells. The Earth is believed to have formed between 4.5 to 5 billion years ago, and the formation of self-assembling protocells is believed to have happened about 3.8 billion years ago. So that's a long time for the formation of these complex molecules. And then around 3.5 billion years ago, the first fossilized prokaryotic cells are observed. And evolution of prokaryotic cells may have probably evolved a lot earlier. Then from prokaryotic cells, the eukaryotic cells evolved. Now, according to the story of chemical evolution, the Earth's atmospheric and surface conditions were entirely different from what we have today. The atmosphere then was a highly reducing atmosphere, not an oxidizing atmosphere like what we have today. So because of this energy coming from bolts of lightning, from ultraviolet light radiation, there was no ozone layer yet in the atmosphere. So very strong radiation hit the surfaces of the Earth then, and you have volcanism. So you have heat coming from the ground. These forms of energies impacted on these simple molecules that led to the synthesis of organic compounds, as evidenced by Miller's experiment in 1953. These compounds accumulated in small pools of water, like soup, which may have been concentrated at various locations in the shorelines or oceanic beds. As the molecules grew and increased in complexity, these compounds were assimilated in self-assembling microspheres formed by amphiphilic molecules such as lipids. These microspheres form the primitive cells or the protocells, which allowed aqueous solution to concentrate inside, and the synthesis of complex compounds were made possible. Evidences strongly suggest that the first enzymes in the protocells were RNA molecules instead of proteins, And the first self-replicating molecules was also RNA molecules. That's according to the evidences we have today. And once this protocells stage was organized and reached, natural selection would have acted on these primitive, self-replicating primitive cells. More rapidly replicating and more successful systems were favored, thereby gradually evolving the first cells, the prokaryotic cells, about 3.5 billion years ago. The prokaryotic cells are distinguished from eukaryotic cells, from animal cells, because these cells are smaller and they have no nucleus. Their genetic material is simply splayed out in a region of the cytoplasm called nucleoid. And they have no membranous structures within the cytoplasm. Sometimes, though, they have infoldings of their plasma membrane, that's all. There's no endoplasmic reticula, nor mitochondria, not even nucleus. So they're very simple cells, the prokaryotic cells. So being very simple, they would have the first to evolve. They would have been the first to evolve. And then later on, followed by the more complex eukaryotic cells. So as you can see here, this summary of events do not only show the evolution of chemicals in living cells, but at the latter two stages, you have the evolution of prokaryotic cells and eukaryotic cells. In the oxygen-poor environment of early Earth, the earliest postulated microorganisms or protocells were probably anaerobic primary heterotrophs that relied on environmental sources of food about 3.5 billion years ago. They were probably heterotrophs according to the traditional theory because the chemical evolution that preceded them had supplied generous stores of organic nutrients in the prebiotic soup or in the pools of water. The earliest organisms would not have been required to synthesize their own food then. But there's another related theory to this, as suggested by Carl Woese. It was suggested that the first protocells were probably primary autotrophs. This was suggested by Carl Woese and Fox in a paper in 1977. They contended that autotrophy evolved in the form of anaerobic photosynthesis. This is a kind of photosynthesis that does not release oxygen and depended mainly on energy from inorganic substances in their environment. Woese also suggested that the earliest metabolism comprised of numerous chemical reactions that were catalyzed not by proteins but by cofactors. These cofactors would have been associated with membranes of the protocells. Cofactors like nicotinamide adenine dinucleotide or flavin adenine dinucleotide. There were no proteins then that were used in this, according to this theory. But whichever were the first primitive cells, the earliest direct evidence of prokaryotes dating 3.5 billion years ago comes from fossilized stromatolites. These are layered rocks that are formed when certain prokaryotes bind thin films of sediment together. Stromatolites are commonly found in shallow marine bays. Fossil data has indicated that oxygen-producing cyanobacteria became prominent around 2.5 billion years ago. And this turned the atmosphere into an oxygen-rich atmosphere within the next 100 years. So the atmosphere then became toxic to most primitive cells in that time, resulting to mass extinction of life forms in the so-called Great Oxygenation Event. We have a lot of names for this event from our textbook in Hickman's Zoology book. An important product of this oxygen catastrophe was the evolution of the eukaryotic cells. The evolution of the eukaryotic cells is popularly described by the endosymbiotic theory. According to this theory, the mitochondria and plastids, that is the chloroplast, were formerly small prokaryotes that began living inside larger prokaryotes. These prokaryotic ancestors of mitochondria and plastids enter the host either as undigested prey or internal parasites. This relationship to the host cell eventually became beneficial for both, hence it is dubbed now as endosymbiotic theory. Over time, the host and endosymbionts became a single organism and its parts inseparable. The picture shown here of the person is Lynn Margulis, who popularized the endosymbiotic theory. The figure shown to the left of the picture of Miss Margulis is the diagram showing the evolution of the eukaryotic cells from prokaryotic cells. The prokaryotes, by which the smaller mitochondrial or protoplast plastid ancestors entered, were large prokaryotes with infoldings of their membranes forming the endomembrane system, which eventually developed a membrane covering around its genetic material, forming the nucleus. And this prokaryote assimilated at some point in time the prokaryotic ancestor of mitochondria, forming what is now the modern day eukaryote. And by another track of events, this mitochondria-rich prokaryote also assimilated the ancestor of photosynthetic bacterium that became the plastid of the modern day photosynthetic eukaryote. That's according to the endosymbiotic theory. Now, for your assignment, to cover some of the points here, I'd like you to answer these 4 questions. By what mechanism might organic molecules have been concentrated with a semi-permeable membrane in a prebiotic world so that polymerization reactions can take place? What characteristic differences are there in molecular structure that distinguish between lipids and carbohydrates? What is the source of oxygen in the present-day atmosphere, and what is its metabolic significance to most organisms living today? And then finally, outline and summarize the evidences supporting the endosymbiotic theory of eukaryotic origins. Submit this as a group work in one short-sized PDF page via email. Thank you and good day.